Okay, in the second part of this lecture, we will talk about enzyme inhibition, specifically on two types of enzyme inhibition, which are the reversible inhibition and the irreversible inhibition. And we will look at how these processes will have effect on the important kinetic parameters such as Km and Vmax. So, what are enzyme inhibitors? So basically, enzyme inhibitors are molecules that are capable of slowing down or causing the loss of enzyme catalytic activity, and they do so by blocking the active site or by changing the structure of an active site. And always remember, protein structure determines its function. So by altering the structure of an enzyme, inhibitors can reduce enzyme activity. So enzyme inhibitors, they are very important in medicine because they can be used as drugs based on their function of enzyme inhibition. So as we mentioned just now, there are two types of enzyme inhibition. It could be irreversible or the reversible inhibition. And under this reversible inhibition, you have more subtypes or subcategories of reversible inhibition, which are competitive inhibition, non-competitive inhibition, and the uncompetitive inhibition. First, we'll talk about the irreversible inhibitors. So they are known as the irreversible inhibitors because they bind irreversibly to the enzyme. And the binding is so strong because it normally involves a covalent bond, which is having a very high bond energy. And it also happens that these irreversible inhibitors will bind to specific amino acid side chain, such as a serine or a cysteine, which contain the hydroxyl functional group and a sulfhydryl functional group. And because such binding is permanent, it permanently inactivates the enzyme, as the active site is now being taken up by the irreversible inhibitors, and therefore it prevents substrate binding. Okay, one notable example of irreversible inhibitor is DFP, which stands for diisopropyl fluorophosphate, and a related family of DFP is the sarin nerve gas, which is the notorious weapon of mass destruction. So what it does is that it will bind covalently to a serine residue in the acetylcholine esterase, which is an enzyme, and when this enzyme can no longer function, it prevents the breakdown of neurotransmitter acetylcholine, and you will lose control of your whole body muscle, including muscles for lungs to breathe in a matter of a few minutes. So normally the victims will die in about 1 to 10 minutes due to sarin gas poisoning. Well, unfortunately, such toxic gas was used in Malaysia 3 years ago. So the half-brother of Kim Jong-un, who is Kim Jong-nam, was assassinated by using this VX nerve agent, which is a type of organophosphate similar to sarin nerve gas. So like sarin, this is an irreversible inhibitor and it is extremely toxic. But in fact, many organophosphates they are used in insecticides such as the mosquito killer or the cockroach killer. Now, if you look at the ingredients of these insecticides, they actually contain several types of organophosphate such as chlorpyrifos, which causes irreversible inhibition of acetylcholine esterase like sarin gas and VX nerve agent. So these are actually the nerve agent for the insects, but they are operate at a much lower toxicity. But at high dosage, these organophosphates can affect human neurological function or even mental development of children. So we better be careful when we want to use these insecticides when there are children around. However, the use of irreversible inhibitor is not always negative. In medicine, for example, aspirin is used to block the action of prostaglandin H2 synthase, which is an enzyme that causes inflammation and fever. And aspirin can also be used to reduce the risk of getting heart attack and stroke. So another example here is penicillin, which is an antibiotic discovered by Sir Alexander Fleming. So penicillin can bind to serine active site within the transpeptidase enzyme, and this inhibits the function of this enzyme, which is to build the bacterial cell wall peptidoglycan. As a result, this eventually kill the bacteria. And as what you have learned before, penicillin is known as a beta-lactam antibiotic as it contains this beta-lactam ring structure. So again, it will bind to the enzyme transpeptidase, and this irreversibly inhibits the function of the enzyme. As such, the bacteria can no longer build any cell wall. But in nature, some bacteria they are resistant to the beta-lactam antibiotics because they can produce beta-lactamase, which is an enzyme that can break down the beta-lactam ring, and subsequently they break down the whole antibiotics. So to counter the action of this beta-lactamase, new generation antibiotics such as augmentin will always comes in a combination of the antibiotic amoxicillin and also an irreversible inhibitor for beta-lactamase such as potassium clavulanate. So with this inhibitor, the antibiotic will no longer be degraded by the resistant bacteria. Okay, there are many more examples of irreversible inhibitors that you can find online. So please read it for yourself and I will not go over them one by one. But you don't have to really memorize everything here, but just to make sure you have a rough ideas of what these things are. And next, we will look at the reversible inhibition, which consists of three different types: the competitive, the non-competitive, and the uncompetitive inhibition. In competitive inhibition, the inhibitor will compete with the substrate for the access to active site. And this tells you why the inhibitor must have similar shape to the substrate because enzymes operate in a key and lock fashion. And when inhibitor is bound to the enzyme, it prevents binding of the substrate and hence you will get no product formation. However, competitive inhibition can be overcome by increasing the substrate concentration until it outcompetes the inhibitor. And this is how you can overcome the competitive inhibitor, which is the guy in black shirt. While you do so by sending more substrate molecules, which are the guys in white shirts. Okay, showing here is the Michaelis-Menten curve for enzyme operations with and without competitive inhibitor. And the first thing to look at is the gradient of the curves. So obviously, with the addition of competitive inhibitor, you can see that the gradient has reduced, which means the activity or the enzyme velocity has reduced with the inclusion of competitive inhibitor. But on the other hand, the Vmax value, which means for the maximum enzyme velocity, remains the same for both cases. Why is that so? So you should know that the competitive inhibitor will compete with the substrate for the active site. So and therefore by increasing the substrate level, we can actually getting rid of those inhibitor molecules and replace them with the substrate molecule. So this explains why eventually it will arrive at the same Vmax value. But also because you have more inhibitor taking up the active sites, and therefore you will need a higher substrate level to arrive at half of the Vmax, and hence you get a higher Km value. So when Km increases, it means the substrate affinity reduces with the presence of this competitive inhibitor. Now, over the right hand side, this plot is known as the Line-Weaver-Burk plot, which is a double reciprocal plot of the Michaelis-Menten curve. So on the y-axis, you have 1 over V versus V, and on the x-axis, 1 over S versus S. But if you look at the y-intercept, 1 over Vmax, it is a constant with or without the competitive inhibitor, which means Vmax remains unchanged. While for the x-intercept, negative 1 over Km, you can see that how this value has become smaller with the presence of competitive inhibitor, which in other words means Km increases. So in summary, when you have the presence of competitive inhibitor, it will increase the Km, which means it will reduce substrate affinity for the enzyme, but Vmax remains unchanged. Okay, showing here is just a joke to strengthen your understanding. So one might ask, how can Ms competitive inhibitor, which is the intruder of the relationship, competes with Madam substrate? That is the perfect match of Mr. Enzyme. Well, the answer is because inhibitor also share similar shape with substrate, which means it is also complementary towards the enzyme active site. As a matter of fact, competitive inhibitor look just like the natural substrates. So in the first example, malonate, it looks pretty much like succinate, which is substrate in the Krebs cycle, while in the antibiotic coming from this family, sulfanilamide, they all look pretty much to the substrate para-aminobenzoic acid. So bacteria will convert this substrate para-aminobenzoic acid into folic acid, which is required in the growth and development of bacteria. So what this enzyme inhibitor does, which is the antibiotic, is to mimic this substrate and prevent bacteria from creating folic acid, and therefore there will be no more growth and cell division by the bacteria. And last but not least, you have methotrexate, which is a drug used in chemotherapy. Now you can look for more information if you want to. Okay, next we will look at the non-competitive inhibition. So in contrast to the competitive inhibition, here the inhibitor will bind away from the active site. And although it doesn't bind at the active site, but it will still modify the active site so that it loses the catalytic power. And as the active site is still empty, it can still bind to the substrate with the same affinity. And therefore, Km is unchanged. But for the enzymes that are already bound with the inhibitor, they will lose function. And therefore Vmax is decreased. Now if you look at the Line-Weaverberg plot, you can see that with the increase in the inhibitor, you get also an increase in 1 over Vmax. So when 1 over Vmax increases, it means Vmax is decreased with the presence of more inhibitor. And the examples of non-competitive inhibitors are like deoxycycline and lead. So it is your homework to find out what are these molecules and what do they do. So how do we differentiate in between competitive versus non-competitive inhibition? So in a normal enzyme binding event, the substrate will bind straight to the active site, but in competitive inhibition, the inhibitor will compete for the same active site, hence lowering the Km. But in the non-competitive inhibition, because the inhibitor will bind to another site away from the active site, so the Km is unchanged. If you look at the Michaelis-Menten curve in competitive inhibition, the Vmax is unchanged while the Km has increased. But in non-competitive inhibition, Vmax is reduced but Km is unchanged. Okay, the last example of reversible inhibition is the uncompetitive inhibition. So what is special is that the inhibitor will only bind to the ES or the enzyme substrate complex. So when there is no substrate bound to the enzyme, the inhibitor will not bind to the enzyme. It will only happen when the enzyme is already bound with a substrate molecule. So in the case of uncompetitive inhibition, both Km and Vmax decreases. And because it will require substrate binding for this type of inhibition to take place, therefore it is less important for drug therapy. Okay, this table summarizes the effect of different types of inhibitions on both kinetic parameters Vmax and Km. For competitive inhibition, the Vmax is unchanged while Km increases. For non-competitive inhibition, Vmax is reduced while Km is the same. For uncompetitive inhibition, both Vmax and Km are reduced. Okay, let's have a thought experiment. Imagine if your hand is an enzyme, then your palm is the active site, while the substrate in this case is a tube. So by flipping the lid of the tube on and off, your hand actually functions as an isomerase. And there are three important parameters to consider: KEQ, Vmax, Km. So KEQ is the same in all cases of inhibition. So what is KEQ? It is the equilibrium constant, which will tell you the ratio of product versus substrate at equilibrium. Now we know that KEQ is a constant with or without inhibition and with or without enzyme. Therefore, it will always be the same. Now, how about Km and Vmax? So let's look at these three scenarios one by one. In the first scenario of competitive inhibitor, your inhibitor will be something that looks just like the substrate. So anything that can fit into a pump can now compete for this active site. And therefore, Km increases while Vmax remains the same. In the second scenario of irreversible inhibitor, so this time the inhibitor will bind permanently to your active site or the pump. You can imagine something like a leaking superglue. So by having this irreversible inhibition, you will get no substrate binding to the active site anymore. As such, Vmax is going to be 0. While in the final scenario, which is the non-competitive inhibitor, this inhibitor will now bind away from the active site. And in this scenario, we imagine that it binds towards the whole hand. And therefore, your pump is still empty and it can still bind to the substrate with similar affinity. And therefore, Chem is the same. Or because of this binding, your active site has been modified slightly. And therefore, you have a reduced Vmax with non-competitive inhibition.